So how many people in the room know that we still have a space program? Awesome. We're going to be friends. Okay. You'd be amazed at how many people, even in Houston, that do not even know that we still have people in space or that we regularly launch things to space. They thought since the space shuttle was gone, NASA was gone, right? Nothing's going on anymore. It's amazing to me. It blows me away that people don't know that. So yes, good. I'm happy to hear that. So the International Space Station is got to be one of the coolest things ever made. This space station is originally started being built in around 1998. Finally, after about 20 years of negotiations and scrambling for money, and was completed around 2000, I think, or no, yeah, 2004. So it's been up there it's been occupied for almost 15 years, and it's about the size of a 747 internally, but the entire size of it is about the size of a football field. Right up here is the space shuttle. If anybody's gone down to LA and seen the space shuttle, it's about the size of a small airplane. That gives you the idea of the size of the space station. The occupied volume is from here and here all the way down. That's where the people are at when you see pictures inside the space station. It goes around the Earth every 45 minutes. There's a sunset and sunrise every 20 minutes. It's going about 17,500 miles an hour. And it's about between 220 and 250 miles up, which isn't very far, right? So it's been up there occupied for a very long time now. It's an unruly teenager, as they say. It's starting to show its age a bit. It's starting to complain like a teenager and have problems that a teenager has. But so far, since almost day one, they have been performing microgravity research in this vehicle, and it's an amazing feat. Not only just engineering-wise, but the fact that there's 15 countries that worked on this. And if you've ever done any work with intergovernmental or government in general, that's an amazing feat. So life in microgravity is like anything else, right? You get up and wash your hair, right? Just like this, no different. You play with your friends, kick around the soccer ball just like you do on the ground. You help some friends move, call you up, say, can we move some stuff? Yeah, sure. Move. Just like they do, just like on Earth, right? You even eat just like you do on Earth. Nothing different, nothing out of the ordinary. Got one more piece to catch here. That would never get old, right? And you can even take selfies in space. I mean, it takes it to a whole new level. Of course, he takes it and looks at it just like we do, right? It's awesome. The life of microgravity is just like on Earth, right? And no, it is not. It may look like it is, but internally, it is a problem for humans. Humans have not lived in a microgravity environment. We evolved on the Earth, right? So there are problems that come from living in microgravity all the way from your eyeballs all the way down to your toes, and that entails such as ocular degeneration from radiation. The astronauts can actually see streaks of light going through their eyes when they're up there as radiation goes through their eyes. There's also problems with pressure building up because the shape of the eyeball changes when it's in microgravity, causing pressure on the back of the nerve. So they actually have blurry vision. Fluids go from your feet all the way up into everything up to your head and your chest. You're literally like a pear upside down. All the fluids come up here. So if you ever see pictures of the astronauts before being in space and during space, their faces usually look really, really puffy, or they sound like they have a cold. And that's because all the fluids have now shifted up. Your kidneys, our filtration rates is completely different than it is on Earth, which causes the next problem, which is bone loss. They lose between 1 to 2% of their total bone mass per month while in space, which is the equivalent of being an 80 or 90-year-old person, having osteoporosis. So it looks normal, life looks normal, but it is far from normal. Microgravity research is not normal either. These are just some basic physics experiments that they've performed on the space station. This is a blob of water hit with a little jet of air. This is actually a little blob of water that's going around a knitting needle he rubbed on his hair, and you can actually see it travels around just like an orbit. You can barely see this down here, but fire doesn't even burn normally in space. There's this little glob of fire that tends to just keep glowing for several seconds, unlike on Earth. So on Space Station, they do everything from human research all the way to astronomy and meteorology. Being so far up, they can actually do a lot of Earth observation as well. It's something most people tend to forget about. For life science research, they've done a lot of vegetable growth. They're trying to grow more and more vegetables to become more self-sustaining. This is actually some lettuce known as outrageous that they've been growing recently. You may have heard about protein crystal growth that's occurred on the space station. These are terrestrial protein crystals that were grown, and these are microgravity, quite a stark difference. They've actually been developing from these crystals alone a muscular dystrophy drug based on the ability to grow these crystals that they could not grow on the ground. Even cells grow differently. On the Earth, we grow them in 2 dimensions, right? And so only 50% of the cells are actually experiencing what they would normally experience in your body. So in microgravity, they can actually grow in 3 dimensions as well and act more like a real cell, or actually act like a real tumor as far as drug delivery or even basic blood flow delivery to a cell. So you get much more realistic type results to growing cells. Almost all the model organisms have flown. This is some of the rodent research that's been going on for the last couple of years. I believe either Merck or one of the big biopharma companies is heavily involved in this now that the resources are up there. This is some Drosophila research that's been going on using multiple different types of models, such as they were looking at heart mutations and basic heart development in microgravity in flies, of which you can grow multiple generations in even a month's time. And C. elegans has done a lot of worm evidence research up there as well. There is a section up there that is specifically designated by Congress as a national laboratory, just as Argonne or Lawrenceburg, as a national lab. And those resources are treated just like a national lab, and you would go through a process to get time on the space station, just like a national lab. It's a little harder to get into, just basic volume-wise. A lot of people want to go there, but it can be done. Research equipment, a lot of stuff that's found on the ground. This is a glove box that's been made specifically for microgravity. They have a -80 freezer, just like you would on the ground. They have incubators, centrifuges, so it's mostly a normal lab. But what was it missing? One basic piece of equipment that is almost in every lab that I've ever been in. It was a plate reader. So this was noticed in about May of 2011, and we selected the SpectraMax M5e. I'll give you the real story of why I got selected. Is the rep came in with it on pulling it behind her. She had it in one of the tow things, I forget what you call it, and she'd actually bumped into things as she walked in. And we immediately saw this and go, "What's in the case? Is it books or something?" And no, that's the spectrophotometer. Really? So we immediately thought, if this thing can survive, obviously this is not the first time she probably did this. if it could survive this, it's going to have no problems in space. And I know from using the same exact instrument when I was working on my PhD, that machine is a tank. I mean, it just keeps going. It's a really well-built machine. So just a bit of history. We selected it in May 2011. In July 2012, it actually arrived to the space station, was launched, became operational in 2014, and actually just came home a couple months ago. Brought it back. So I don't know, everybody works on here. I'm pretty sure you all know what this looks like, but just in case there's not, this is what the M5e looks like normally. So we go through a process of what we call flyberdizing. And this is preparing something to fly in space. And it's kind of a mix of flying and hybridizing, funny word. And so we do things that we need to do for it to at least survive somewhat in a microgravity environment. So you've probably seen this next picture too, but that's what it looks like now. Basically, this is what's called a mid-Dec Locker in the back, and this is actually the SpectraMax itself, and it actually goes all the way to the back of the locker. It's about this big. It's about that big. And this mid-Dec Locker thing is a standard that was formed back in the shuttle area, and that's what's called a mid-Dec Locker, because these were actual compartments that were formed in the mid-deck of the space shuttle, and it just became a standard size format. That's been used over and over again and now used on the space station. So your mid-deck locker fits in these little compartments in what's called an express rack. The rack itself has all the gases and fluids and electronics and everything you need to take care of whatever in these boxes. This is actually about the size of what an express rack is. You can see the crew getting a delivery of one up on the space station back when they were first assembling the space station. And they go into the actual all four walls of the space station modules themselves. So you can actually work in all directions around the space station. So we had to first be able to take the plate reader itself and put it into that size format, which actually wasn't believably hard as it sounds. The other reason we put it in a metal box is to decrease EMI, straight electromagnetic interference on the space station is a big problem, and it's one of the first safety requirements NASA requires from us. Is we don't want your instruments screwing up the space station and blowing it up or anything like that, which wouldn't be good. The other thing we had to do is we had to use a power conversion from 110 to 28, normal 28 volts, which is found on the space station. The other power safety problems or issues that we had that NASA requires are such things as fuses and extra circuit breakers. We added a mini PC that ran an emulated Windows XP and that ran SoftMax Pro. And we had a basic crew, very easy crew interface with a touch screen. The usual procedures that were followed is the PI would come up with their experiment, prepare the plates, either put them in 4C or minus 90, minus 80, whatever you want, load the protocol on a USB stick. We deliver it to NASA. They put it on a capsule, send it up to the space station. And depending on what the procedures for the experiment needed, such as if the crew needed to take them out, the plates out and thaw them, did you need to take different reads every day? Everything was different. So every procedure was different, so I didn't list them here. So it really varied on what the PI wanted. This is the face of the plate reader. Here's the touch screen that I had talked about. We have the various different buttons that we had to add in order to operate it. So we have a standard power connection, on and off switch, 15 amp fuse, the jack for the USB. And then this is a cover where the actual plate is inside the plate reader. This is what the touch screen uses. We kept it easy. It had to be quote unquote dummy proof so that the crew really had a minimal interaction with it other than putting the plate in, push and go. Yeah, so the USB was loaded with the program. Upon it was put in, the USB was plugged in, turned the machine on, and it would automatically load we had made it so the software would automatically load the program. Put the plate in, hit close drawer, and scan plate, and it would go. This is what, it's really hard to see, but this is just what the little door open, and this was just to keep anything in there from actually getting inside the plate reader while it was running. The only modification we had inside for the carriage and the microplate carriage was a little piece of cellophane, and that was just to keep the plate from sitting inside before the drawer actually closes and the little latch closes, sets it against the carriage. You had to have something in there to actually keep the plate from floating away. So a little piece of cellophane worked perfectly. It kept the plate from floating away. This is a little bit complicated, but this is looking from the top of the microplate reader, and this is where that little touch screen is over here, and this would be the front. So the lamp would be back here if you're familiar with the guts of the plate reader. Everything from here and here is straight out of the box. We literally just took the case off the plate reader itself. Below here is where the carriage was that I just showed, and then right here is the little mini PC that we used. Over here, just the voltage regulators, connections, and there was another voltage regulator on the top that we used. This is from the side, from one of the other sides with all the voltage regulators that we had, a couple fans for cooling. And that was basically it, not a lot of modifications. We really just took this instrument and minimally, I think 90-95% straight out of the box, commercial off the shelf, as we call it. We've had 3 projects last year that flew. One is called Apex 2. There was a project from Stanford Burnham called MicroGRX, and another one called Project Mercury / SideCheer. Apex 2 was called the Advanced Plant Experiment. It was from a PI named Tim Hammond from Veterans Affairs Office in Durham, North Carolina. He was growing yeast with GFP and M-Cherry fluorescent proteins and looking at gene expression in microgravity. Perfect for the plate reader. It had 100 reads over 18 hours, and he had 10 plates that were spotted with the yeast. The yeast went up in 4C. The crew would take them out of the fridge, let them thaw for a little while, and then put them in the plate reader and hit go, and it would sit there for 18 hours. That was the plan. We had the first run after about 5. Actually, it ran once. The crew had already taken that plate out and put the second plate in, and we had sent we got the data downlinked from station and sent it to the PI. And within hours, the PI calls us and there's something wrong. So we looked at the data, and about 5 hours into the data, the fluorescent signal seems to drop from about 5,000 to 25 to 300 RFU, all 384 dots that he had. The temperature was also between 38 and 40 C, which is not normal. And but it appears that the data continued to be collected all the way into the run. Here's the data on just 3 of the wells. If I put all the wells on here, it would look a mess. Collecting data very nicely up until about 4 or 5 hours, and then it just drops to this. It didn't go completely to 0, but it went, like I said, between 25 and 300. It varied on which plate. I don't know. Some of the wells, not all of them, had these little spikes in them of fluorescence, and then dropped. So the one thing that the PI had mentioned is one of the PMT died. We were surprised at the high temperatures that was inside of it. So we set up the ground unit. We have an identical ground unit on the ground, and tried to stress it out. And I put the plate reader, our ground unit plate reader, in my garage in August in Texas, and ran it for 36 hours. And the computer, the little mini PC, failed, but the plate reader did not fail. Exactly. So it was. I mean, you couldn't even put your hand on the box. It was that warm. So temperature was not it. I'm very certain the temperature was not the problem. I'm convinced, though the PI has yet to say this, that I think his yeast died. I think they just died and the signal died. Right? That's what makes most sense to me, because of the heat, because it was so warm. It was yeast. The yeast is happy at 30. This got up to about 40, which makes bread at that temperature, I think. So the yeast died, and so we weren't able to continue on with this experiment, unfortunately. We will be flying his experiment again later this year when we build the next version, which I'll get to at the very end. Project Mercury was a really interesting. Sorry, go ahead. Correct. Yeah, and we know now what it is, but at the time. So the original design of the plate reader was to do one run, 5-10 minutes, and be done. We had not thought of people actually using it as an incubator, putting it in for 18 hours and letting it go. So cooling was not a big initial design feature, which we now know that we're designing into the next phase. So they wanted to, I still don't know how they got mercury out of this, but they're comparing pretty much the growth rate of DNA, DNA characterization. They're looking at microbial growth of various different microbes. John Eisen from UC Davis. And they just wanted to look at how different communities grow on an agar-based plate, 96-well plate. Just basic absorbance, 1 read per day for 5 days. We actually they wanted to do long runs, but I was afraid it was going to cook their samples as well, so we just got 1 row and run per day for 5 days. And that predicted that would give them a nice growth curve, and it did a good job. Now, where these microbes came from was really cool. So this is a group called SideCheer, and they are a group of cheerleaders, real cheerleaders that have PhDs, or used to be cheerleaders and now have PhDs. And what they do is they go out and they encourage STEM activities for especially women in science. And they went all over the country collecting microbes with microbial swabs from everywhere from the Liberty Bell to a lot of different various sports arenas to even the bottom of Buzz Aldrin's shoe. So they had this huge collection of microbes from across the country, especially sports arenas, which seemed to be really cool. So they came up with the idea to take the sports arena microbes, especially ones that were significantly different from each other, put them on a plate and compete them, the ones in microgravity with the ones in the ground. And this is right around Super Bowl time, right? So perfect, great idea. This is what the plate looked like, nothing special, just bacteria on a plate. This is the box that it went up in, and they went up frozen at -80. I keep mistakenly say -90 because for some reason the space station freezers are -95. And this is the data they came up with all sorts of categories, like best sprint, the fastest one, and they have like 12 different categories for it. It was a great public outreach event, showing essentially that microbes aren't as bad as you think they are. They're everywhere, and a great encouragement for girls and women in science. The last one was initially supposed to be just a test of whether we could do fluorescent polarization in microgravity. Nobody had ever done it. Now we had a plate reader. Let's try it. Would it work? Who knows? A couple PIs from Sanford Burnham in Orlando, Florida. They used just basic biotic conjugate with an antibody, the biotin, and for absorbance they had food coloring. They had 4 plates, UV vis, fluorescence polarization, fluorescence intensity, and they had a total of 6 runs. I don't have any of their data. They're still working on it, but the initial data that I'd sent them and they had graphed said it looked fine. There was no problems. And this was run after the, I forgot to say that, so these 2 runs that I just showed you were done after the anomaly that we had with the Apex run. So we had to do a lot of cartwheels and appeasement to NASA to allow us to run our plate reader and pretty much show them that this was not going to blow up the space station. It's not going to go into a crazy heat cycle and melt, a hole in the wall. I'm not kidding. Everything you can think of. So once we proved that it was safe, we actually did a test run. And they allowed us to do these 2 final runs. It ran fine. So we're even more flummoxed. What happened with the initial experiment, we still don't know. Everything from yeast died or it took a radiation hit was another thing that was brought up, since the radiation in space may have just caused one of the PMTs to crash out. I don't know. We still do not know. So for the fluorescence experiment that was done, their 384 well plates just filled to the top and then sealed. For his, it's just a spotted yeast plate. There's just 384 spots of yeast, and it was reading from the top. It's got one of the plate adapter in the bottom. That's the only requirement, is just to fill it to the top with no air bubbles of the plate, because the air bubble will obviously hurt your data. So the plate reader came home, I think it was February or March. We call it a success. We had 22 runs and 1 anomaly. I think that's a success for this plate reader, for the first of its type ever done. With minimal design on it. We didn't really do much to it but make it safer to be on the space station, right? Immediately when I got the plate reader back, I ran the Apex program again, the same exact unit, just to see what would happen. It ran fine. I had no problems with it. The data returns are normal. I actually was able to pull the error log off it with some help from Crystal's here. Crystal Berger's been awesome. She's been helping me with via email with questions. She's been really, really, really awesome. I literally sent her, we were working on something, the next generation one. I sent her a picture with a cord that was inside the plate reader, and I said, "What is this?" She said, "Hold on." A couple hours later, she got the answer. I mean, she's been really good. I just wanted to mention her and thank her. She actually told me how to grab the error log that was off the plate reader. And there were no errors that corresponded to the PMT failure, none at all, or any type of failure. There was actually 3 errors, but they had nothing to do with any sort of PMT failure or fluorescence. So we do not know what happened with the Apex run at all. This is how it came home. It splashes down into the Pacific Ocean, just off the coast of Mexico. Actually, probably landing in the ocean is probably softer than the parachutes pulling out, which is pretty good. So it took some G's, and we're working on getting some validation placed to see. Is it still in spec? Is it out of spec? How bad just after being in space for several years, how bad out of spec it is? No. Just out of curiosity, how it is, because we're gonna reconfigure it and fly it again. Yeah. This is a little bit stronger than hitting it with the carrier. Launch is pretty gentle. Launch maybe max of 3 Gs, and that's pretty soft. It's not for very long. But this is a kick in the pants. So speaking of the next version, I call it this Plate Reader Strikes Back, the Plate Reader 2. We're definitely working on how to definitely negotiate the air within the plate reader itself. We have a plan with some just fans, really. We've already had a basic preliminary setup with just fans, and we were able to significantly cool it down to the normal temperatures without it getting warm. It just wasn't initially designed to dump heat. It was only made to do short runs. We're putting in a brand new mini PC and upgrading it just to something new. We're also using the newest version of SoftMax Pro, which is actually, I think, one of the best versions out there. We're removing the touchscreen. One of the problems with the touchscreen is we didn't integrate any sort of software to, if you hit go or you hit something on mistake, that it would not stop it. You had to turn the machine off again, so that's a big problem in microgravity. We actually had one of the crew members say that, hey, could you next time not have this, or have something to where if I hit the screen on accident, it doesn't just go. So crew actually gave us a lot of feedback, which we've implemented as well, just basic design. The only thing that'll be in front is the LCD screen that was on top. We're now going to put that on the front, because that was another problem when we did have that initial anomaly, is that we had no way of seeing the LCD screen to see if it was throwing an error. It just had, you know, that would have been of value to us just to see an error code to see if anything was wrong. But we didn't have the ability to do that. Probably the coolest thing that we're now able to put into the machine is to teleoperate it. We'll be able to run this machine from the ground. So all the crew has to do is put the plate in. The big bottleneck right now is crew time. There's only 6 people up there, 3 of them which are working for the National Lab, and they have to sleep and do other things, unfortunately. And maintenance of the space station, really. They spend a lot of their time just keeping the thing running, fixing the toilet, believe it or not. Basic stuff you would in your house, right, an old house. It's amazing. They still, on average, do about 30 to 40 hours of research a week. But that is, as Ron had pointed out, is significantly increasing, which is a good problem to have. And I think a first ever in microgravity research is people are realizing this is a resource to use for research. That's an opportunity that was just never ever there before in anybody's lifetime. And this is launching in fall of 2015, so less than a year. We're fixing it, turning it around, and we'll have it up at the end of the year. Again, just quickly, just something we also flew, just a couple of our other products just to show you. We have a little off-the-shelf handheld microscope that just went up. This is actually the 3rd version of it. It's about 10 to 240x. We built this stand, and everything is commercial off-the-shelf. We built a little holder for a 96-volt plate microplate holder, so if anybody wants to send samples up in a microplate, we can use this. This arrived just last month. We're actually building a robot. Speaking of crew time being a bottleneck, one of the ideas we had is, could we just have plates in an incubator, have a robotic arm, and have a microscope in it, so the robot will just grab the plates, put it on the microscope, look at your plates, and then put the plate back. Just something that simple, which would be simple in a lab, is a lot up on space station, again, because of crew time, just something very basic like that. So we're building this robot arm, which will, I think, help a lot as far as the ability to actually do more science over time, because this thing will work 24/7, just like a grad student. So we're hoping that this will, we're looking at the end of the year for this to fly, which it's going to be the first of its kind. There's never ever been anything up there. There's that big robot that's up there, Robonaut. But this is specifically to do science, and there's nothing like that up there. It's a very common thing in a lab now to have robots, right? And there's nothing up there like that. So just wanted to say thank you. Again for Molecular Devices. I can't even see it. He actually fogs up the window and draws a little smiley face. Anyways, I just wanted to thank you guys. Molecular Devices have been awesome to work with in helping us design this. In the next generation, and we're always looking for more instruments to put on the space station. There's a lot up there, but I think the space station can be made better. I think we can make more instruments to do better science, more science. And thank you.